It should be starting. Okay, it's going. Great. So thanks everyone for joining us. I just have a few intro slides before I turn over to our speaker today. So I think most people are familiar with both HESI and with eSTAR, but just want to take one slide to show our diverse portfolio. All our topics are related to new systems technology, a lot of these either catalyzing new science or looking for biomarkers. Here's just a high-level overview of our portfolio with some nice highlights. So our carcinogenomics group and our FFPE group have recently come out with new publications. And just want to highlight, we have some new projects that are forming, the cell painting and error-corrected sequencing. So if folks are interested in any of those, if they're involved in one but want to be on another or are new to this, please just let me know and we can talk about any of those. Just some logistics and protocol for teams. So please mute yourself unless you're speaking. We're going to have Subham present and then at the end have time for discussion. So I ask everyone keep their selves mute. At the end, when we have that discussion, please feel free to raise your hand or come off of mute to ask a question. Throughout the talk, you can also put things into the chat and people can like things in the chat so we know to go to that comment or question. There is an ability to do closed captioning. So how to do that is in that picture there. And I asked once we get into the actual presentation for everyone except Subham to turn off their camera. If you have any technical issues, you can email myself or Siddharth. Okay, just one last plug. If you have another idea for a webinar, please just go ahead and email me of things related to our topic. And I'm now going to introduce our speaker. I'm really excited about this one. So Subham Dasgupta is going to be giving the talk today. He's been one of my mentors in my career and is a fantastic scientist. So Dr. Subham Dasgupta is a molecular toxicologist working currently as a postdoc fellow in Robin Tangway's lab at Oregon State University. He received his bachelor's in human physiology and master's in environmental science from the University of Calcutta, and his PhD in marine science at Stony Brook University in New York. He's trained as a developmental toxicologist, primarily using zebrafish embryos as a model, and has worked extensively with flame retardants during his postdocs, both at UC Riverside and Oregon State. And he's just accepted as a job as an associate professor at Clemson University and will begin this fall. So everyone, please welcome him. And I'm going to stop sharing. And Subham, you can start sharing.
Hope it's been shared.
It's going. I saw you click the button. It seems like there's a few seconds delay on that, but we see the slides. Yeah, there it goes. Perfect. All right, go ahead.
Okay, thanks so much, everyone. Thanks for inviting me here today, Connie and everyone else. Thanks for attending. My name is Subham Dasgupta, and as Connie mentioned, I am currently a postdoctoral scholar in Dr. Robin Tangway's lab at Oregon State University. And I have a number of different projects, but one of the projects is to look at the toxicity of flame retardants using zebrafish as a model. So today's presentation will be based on how we use phenotypic anchoring as well as combined mRNA microRNA expression profiling to understand flame retardant chemical toxicity networks. So a big part of my research relies on understanding what are the environmental determinants of health issues. Within our everyday life, we are exposed to limitless cocktails of pollutants, and many of these pollutants contain bioactive chemicals which can interact with biology and cause adverse health effects. And therefore, we need to be able to identify these bioactive agents and study their health risk before they can cause long-term effects in humans. And the goal is to understand disease mechanisms and create therapeutic and preventative pathways. This includes facilitation of drug development, dietary counseling, lifestyle changes, but we also need to model toxicity for emerging chemicals in order for them to prevent creating adverse health effects in the future. And the goal of all of this is to design safer alternatives as well as engineer remediation strategies for the toxic chemicals that are currently in commerce. The best strategy to achieve this goal is to detect immediate or early responses a chemical elicits as a response to biologically relevant tissue doses. So for any chemical during right after exposure, they interact with biology to cause these immediate or early responses. But these responses are often molecular, and unless specifically studied, can often go undetected and cause long-term effects, or might combine with other stressors in life to cause later life or chronic life stage impacts such as neurodegenerative diseases. This is particularly important for developmental health since early effects may translate into irreversible lifelong effects. And therefore, we need to be able to identify these chemicals in order to prevent adverse health outcomes for humans. And one of the classes of chemicals, as I mentioned, that I worked with are flame retardants. So what are flame retardants? Flame retardants are chemicals that are used in consumer products to slow down spread of fire. They are used in a number of everyday products that we encounter around us, as you can see from the figure here, such as the chairs that you are sitting on, electronics, car seats, carpets, gym products, and so on. So in a lot of other products. But why are we talking about their health effects? This is because many of these flame retardants are what we call additive flame retardants, which means that they are not chemically bound to their end-use products, and then they can leach out into the indoor environment and can be integrated into the dust and lead to human exposures either through ingestion or through inhalation. In fact, measurable levels of number of different flame retardants have been detected within human biosamples. Flame retardants can also create exposure disparities, and studies have shown that people of color and low-income communities who may not be able to buy new furniture or cars can be chronically exposed to high flame retardant levels that are leaching out of from these older products. So this slide lists some of the exposure assessment studies for flame retardants. Just to reiterate from the previous slide, they have been detected in measurable and even high levels of concentration within household furnitures. Um, on commuters commuting long distances in cars, it has been shown that flame retardant exposure can increase as a result of commute time. Uh, they have been detected in gym products, a lot of foams that we used in gym mats. Um, they have been detected in baby products as well. In fact, one of the earlier toxicity of flame retardants in the 1970s and 80s was associated with baby clothing or children's clothing. There's also occupational exposures for flame retardants for firefighters because of the nature of their job. And finally, many flame retardants, including the more persistent halogenated ones, can be present in food through bioaccumulation in the food chain. In fact, due to their potential toxicity, a previous group of widely used flame retardants called the PBDES were phased out and replaced by substitute flame retardants such as the organophosphates in 2005. And since then, the metabolites of this organophosphate flame retardants have amplified within human biosamples. So this is, you can see, since the previous PBDEs were phased out and the organophosphates came into prominence, the metabolites of organophosphates such as diphenyl DPHP and BDC-IPP have increased in humans, in urine samples from humans, almost 100 times between 2005 and 2015. And this suggests that there is an increase in exposure over this time to these organophosphate flame retardants. Naturally, the concerns for toxicity arises, and studies have taken multiple different approaches to understand adverse health effects of flame retardants. Epidemiological assessments, as these two studies shows, have shown that flame retardants exposures can be associated with neurological or reproductive outcomes. But researchers also have used an array of different model organisms, including zebrafish as a model. This is a study that was led by Connie when we were working at University of California, Riverside. But there's also other different model organisms, such as mouse models, C. elegans, as well as in vitro and in silico approaches to understand their mode of action and better predict their health effects. However, the challenge with studying flame retardants is that flame retardants do not belong to one class of chemicals, but belong to several different physicochemical classes, as you see here, consisting of many compounds per class. And many of these chemicals are manufactured in several thousands of metric tons per year and used in end-use products. Therefore, their toxicity from their exposure need to be evaluated collectively, and currently presents a challenge because because of such a diversity in their classes. But we need to be able to understand if the adverse effects are derived from a specific class of flame retardants. Which brings me to the objective of this work. Our work achieves this goal by using zebrafish as a screening model for a library of 61 different flame retardants belonging to diverse physicochemical classes. We also furthered the knowledge of the molecular targets of these flame retardants by pairing mRNA and microRNA sequencing for select flame retardants based on the initial screening. Our hypothesis and goal is that this will give an idea of the biological targets of flame retardant based on their chemistry. So why zebrafish? Zebrafish is a terrific model for high throughput detection of chemical bioactivity by studying phenotypes. This is possible since they have external development and have transparent embryos where the developmental phenotypes can be tracked in real time either through light microscopy or in transgenic models through fluorescence microscopy. As you can see here, a big advantage of zebrafish is that their development is rapid. So from a single cell zygote stage to the onset of organogenesis, it takes less than 24 hours. Furthermore, they have a high fecundity, which increases the capacity of chemical testing. And taken together, all of these traits of the zebrafish model enable it to be used as a biosensor for rapid detection of chemicals that target biology and to prioritize chemicals for further tests as well as regulatory decisions. From a research translation perspective, zebrafish is also used as a translatory model. This is because zebrafish has a high similarity, more than 75% similarity with the human genome. And what's important to know is that particularly in this embryonic zebrafish model, during the process of development, the full molecular repertoire is active. And this includes many of the disease-causing genes, which also have important roles in development. And therefore, by exposing chemicals to these early life stages, we can not only study the impacts on development, but also we are able to predict disease potential later in life and study how these stressors modify these genes. So because of these innate advantages of zebrafish as a model, it has been used widely in biomedical research, including regenerative biology. Zebrafish-based reverse genetics and gene editing is common now, including technologies such as CRISPR-Cas9. Zebrafish has been widely used for behavioral screens as well as chemical and genetic screens, the chemical screen part will form a large part of my talk today. Zebrafish has also been used in cancer biology as a xenograft model. And also, overall, zebrafish has been widely used as human disease models. In fact, more than 500 adult diseases have been studied using zebrafish as a model. So using these advantages of zebrafish as a model, as a screening and translational model, we screened a library of 61 flame retardants belonging to diverse physicochemical classes, including, as you can see here, there's esters, there's ethers, there's phenols, there's also halogenated and non-halogenated flame retardants within our library that we screen. So we try to maintain a balance of this library, and this library was acquired either from EPA or Sigma. Our first aim for this project was to perform a developmental screening of 61 different flame retardants and identify bioactive flame retardants. And aim two of this project is to base our it was based on this developmental screen and identify 10 different flame retardants and perform mRNA microRNA profiling of these 10 flame retardants to understand their molecular regulatory network. So I'll talk about the first aim over the next couple of slides. This involves the developmental screening of the flame retardants, and this part of the project was mainly led by Dr. Lisa Truong in our lab. So within this project, we did a bioactivity screening of 61 flame retardants. Our exposure paradigm is for any chemicals, including the flame retardants, is that we typically initiate the exposures at 6 hours post-fertilization, which is approximately the gastrulation stage of the embryo, and our exposure period lasts for 5 days. At 5 days, we do an observation for developmental screening phenotypes, including mortality, whether the embryo is dead or alive. We do a range of morphology phenotypes, including 22 endpoints, such as bent axis, edema, jaw and snout defect, or caudal fin defects. And all of these morphology endpoints indicate that a chemical affects biology to some extent. In addition to that, we use this instrument called the Viewpoint Zebra Box to do two kinds of behavioral screening in the larvae. So the first screening that we'll extensively talk about today is the larval photomotor response, or the LPR. So in this screening, or in this assay, we expose the embryos to alternate cycles of light and dark, and record their behavior in response to these light cues. We also have the larval startle response, or LSR, where we measure the larval behavior in response to an acoustic startle. So for our experimental design, we used 96 well plates with 1 embryo per well. For this particular project, depending on the chemical, we tested 10 different concentrations and normalized our data to a 0.64% DMSO. We used 2 replicate plates with N of 16 per plate, with a total of 32 animals per concentration, and again measured the 22 morphological endpoints as well as the 2 behavioral assays or endpoints. So this slide represents the results from our developmental screen, and I know that these figures are really hard to read, so I'll try to walk you through this figure. So what you see at the top is the 61 different flame retardants. In the bottom, if you see a red circle, that means that the flame retardant was positive for a morphological phenotype. If you see a blue or a yellow circle, that means that the flame retardant was positive for a behavioral or an LPR phenotype, blue meaning hypoactive and yellow meaning hyperactive. And the size of the circle denotes the intensity of the effects. So from this figure, what is very apparent is that the flame retardants are really bioactive. So many of these flame retardants elicited effects or were hits for either the morphological or the behavioral, or both morphological and behavioral endpoint. Now, if we focus on the asterisks and the pound signs on the top, the asterisks indicate the select PBDES, which are the older generation flame retardants, and the pound signs denote select OPFRS, which were the substitute flame retardants. The screening data that shows that the PBDs and the OPFRs overall were not very different in terms of bioactivity. Although if you focus here, BDE47, which is a legacy flame retardant that has been well studied for its toxicity, was less bioactive compared to most of the other flame retardants, including other PBDEs and OPFRS. So within this slide, we show some data from our lab and comparative data from other labs that indicate that zebrafish was a sensitive model for detecting flame retardant toxicity. So within this figure, what you see on the y-axis is the lowest effect concentration, the points of departure of the benchmark doses for different flame retardants in different studies. The study from our lab is denoted in green by the word Tangway. What you see here is that for most of the flame retardants, the zebrafish model was able to detect bioactivity compared to many of the other models that are used for screening and study flame retardants, which gives us the confidence that this is a great model to detect flame retardant bioactivity. So based on this, I will now move on to the part that I was primarily involved on, which uses paired mRNA and microRNA sequencing of 10 selected flame retardants. And this work was published last year in Frontiers of Cell and Molecular Biology. So like the initial screening, the 10 selected flame retardants belong to diverse physicochemical classes ranging from ethers to OPFRs to PBDES. Based on the screening data, EC80s for these chemicals were calculated, and the mRNA and microRNA sequencing was done at this EC80 concentration. For some chemicals, there were no EC80s because we did not see substantial phenotypic morphological effects. So for these chemicals, the exposures for mRNA and microRNA profiling were done at a limit concentration of 85 micromolar. So these are the actual concentrations that were used for the molecular profiling. So this is the data, the screening data from the selected flame retardants based on the previous study. And what you see here is that not only did we choose the flame retardant based on diverse physicochemical classes, but they were also chosen based on diverse bioactivity profiles. Some of these flame retardants that were chosen were hits for both morphological and behavioral phenotypes or LPR phenotypes. Some of the flame retardants, including BDE47, were hits for only the LPR phenotypes. Some of the flame retardants, such as this organophosphates, were hits for mainly the morphological phenotypes, as you see here, whereas some of the flame retardants, such as TCEP and TCPP, were not hits for any of the phenotypes. Now, you might wonder why did we choose these two chemicals if they weren't hit in our bioactivity profile. Our intention was to also, in addition to anchoring the molecular responses to the phenotypes, our intention was also to see if we could see any molecular responses in absence of this any apparent phenotype. So these are chemicals that might lead to toxicity later during the life stage, but we did not see any phenotypes at 5 days. And we wanted to see if there are any molecular responses that are underlying the absence of any apparent phenotypes. So we have been talking about gene expression, and mRNA sequencing is a commonly used technique now, but why are we studying microRNAs? MicroRNAs are small, typically less than 100 nucleotide non-coding RNAs that can bind to 3' untranslated regions of mRNAs. And they can act as post-transcriptional modifiers of mRNA functions, either by repressing translation or by degrading mRNA. MicroRNAs are conserved across species more so than mRNAs, and over the years, over the last few decades, they have been shown to be really important for diseases and development. However, their role in toxicology of these microRNAs has not been well studied, and the objective of this project was to understand if microRNAs regulate gene expression changes in response to flame retardant exposures. So for this part of the project, for this microRNA mRNA sequencing, our exposure period was 2 days instead of 5 days. So why are we choosing 2 days? The 2 days exposure period was chosen to capture the genetic signature prior to seeing any phenotypic effects or mortality. Uh, as I talked about in one of the introductory slides, that we should be able to detect chemical or bioactivity responses prior to seeing any actual effects, actual disease effects, and this choice of 2 days of exposures is aimed towards that, where we are seeing the molecular signal prior to seeing any phenotypic signal. This also ensures avoidance of effects due to systemic toxicity or grossly deformed fish. So at 48 hours, we collect samples, we do RNA isolations, and we do small RNA and mRNA extractions, followed by small RNA sequencing for microRNAs and mRNA sequencing on an Illumina platform. We followed this up with differential gene expression, gene ontology using Metacore, and we then performed assessments for mRNA microRNA interactions, as well as transcription factor analysis using a platform called TFMIR. So what we see here are the gene expression and mRNA, microRNA expression profile patterns for the flame retardants aligned with their bioactivity profiles. As you can see, we see all the flame retardants, regardless of their classes, led to most of the flame retardants, regardless of their classes, led to large-scale disruptions of both gene expression as well as microRNA expression. But what was also interesting is that even flame retardants without any phenotypic effects, such as TCP and TCPP, which are chlorinated phosphate, showed substantial gene expression changes and to some extent even microRNA expression changes. And this suggests that there can be underlying molecular changes in absence of phenotypic changes, but it also suggests that it might be difficult to group these flame retardants based on their gene expression and chemical class profiles. On the right side, you see the expression of top 10 genes and microRNAs across for each flame retardant, plotted across multiple different flame retardants. Again, what you see here, that in spite of these chemicals belonging to diverse physicochemical classes, there were several common genes and microRNAs across the flame retardants profiles, regardless of their physicochemical class. When we did gene ontology assessment, the gene ontology suggested that in addition to other classes that were disrupted, nervous system development was the most frequently disrupted pathway dysregulated across the flame retardants. This was true for both mRNAs and mRNA targets of microRNAs that were disrupted. We also identified in concordance with this, the nervous system development across a number of different flame retardants. We also identified several neurodevelopment associated genes, which you see in panel C here, that were disrupted across multiple flame retardants. And many of these genes play a role in neurogenesis, calcium homeostasis, neurotransmitter synthesis, as well as glutathione regulation, all of which are important for nervous system development and function. And all of this suggests that nervous system might be a potential target for these flame retardants, but this doesn't discount the other pathways as well that are disrupted across many of the flame retardants here. So we then performed a regulatory network analysis between mRNA and microRNA targets of flame retardants. So again, this chart is really hard to read, but I'll guide you through this. In the bigger red nodes, you see the different flame retardants that are plotted here. The bigger yellow nodes represents microRNAs that were disrupted across 5 or more flame retardants, whereas the blue nodes denote the microRNAs that were disrupted in less than 5 flame retardants. And the text in the arms denote the different genes that these microRNAs are predicted to regulate. Overall, the analysis revealed an extensive set of common interactive networks between microRNAs and genes for multiple flame retardants, indicating common molecular targets and regulatory patterns. In fact, refining the data to a few selected microRNAs, we plotted the expression of the genes that these microRNAs are predicted to regulate across the flame retardants. What we see here is that the microRNAs regulate a diverse set of genes that are that in turn regulate several biological processes, including apoptosis, transcription factors, neurological pathways, as well as other pathways such as inflammation or angiogenesis or other pathways. Overall, this data suggests that microRNAs should be an important consideration in studying flame retardant toxicity. This also suggests that microRNAs do not regulate specific pathways in flame retardant toxicity, but regulate a number of diverse biological functions. However, what's important to know is these are all mostly predictive mRNA-microRNA interaction, and the knowledge gap exists in functionally confirming the role of microRNAs in flame retardant-induced gene expression changes. Based on the expression data, the gene expression data, we were also able to compare the signatures for several flame retardants, the molecular signatures for several flame retardants. Here you see the comparison between TBBPA and TBBPA-DBP, the DBP being a transformation product for the former, and also the DBP, the ether, is used as a replacement flame retardant for TBBPA in several uses. What you see here is between the parent and the transformation product, the phenotypic profile is distinct. The TBBPA disrupts only more of the morphological phenotypes, whereas the DBP only disrupts the LPR phenotypes. And this is also reflected in the gene expression profiles, where there are minimal similarities between TBBPA and TBBPA-DBP. Gene ontology assessment shows that these two related flame retardants have distinct clusters of molecular targets. For example, if you see the gene ontology assessment for DBP consistent with it disrupting the behavioral phenotypes, gene expression shows a large-scale effect on nervous system development. Similarly, similar to the prior slides, we were also able to compare the older generation to the newer generation flame retardants. So in this couple of slides, we compared PBDE, BD47, which is an older generation flame retardants, with the organophosphate flame retardants, TCEP and TCPP. Both of these are chlorinated phosphate esters, and IPP and TPP, which are both aryl phosphate esters. And based on this, what we show is that even amongst the OPFR group, the aryl phosphates, IPP and TPP, were far more bioactive than the chlorinated phosphate. This suggests that bioactivity profile can widely vary even within the same major group of flame retardants. What's also important here is that the BD47 only showed behavioral or LPR effects. When we look at the gene expression profile, it becomes more complicated. Here you see a comparison between BD47 and the aryl phosphate, and the BD47 and the chlorinated phosphate. What you see is that in spite of IPP and TPP having similar phenotypic profiles, we see that their gene expression signals are very different, where IPP is much more potent than TPP from the point of view of this molecular signatures. Similarly, between TCEP and TCPP, remember that none of them induced any apparent phenotypes, but TCEP led to large-scale gene expression changes. In addition to that, we also see that several of the gene clusters were common to all of the three classes, either the PBDES or the organophosphate flame retardant, suggesting that it's challenging to bin them according to their physicochemical classes. And therefore, we see that there are commonalities in gene expression profiles between different flame retardant classes, but also there are large-scale differences within members of the same class, such as IPP and TPP or TCEP and TCPP. Based on this, again, the summary is that it's still challenging to bin the adverse effects of different flame retardant classes based on their gene expression profiles and their chemical classes. So similar to the mRNA and microRNA analysis, we were also able to create a network of disrupted transcription factors. Within this figure, what you see are the flame retardants represented in the larger red nodes. The transcription factors that were disrupted across 5 or more flame retardants in this bigger black nodes, and the rest of the transcription factors in the yellow node. And as you can see here, this also showed a large number of common transcription factors disrupted across flame retardants, including transcription factors such as retinoic acid receptor, vitamin D receptor, and retinoic X receptors, which are known to play a role in development, but also in later stage diseases such as cancer. And therefore, this shows that we are able to predict an impact on these molecular targets using this model. And finally, as a special case, we also found a number of cases where a gene regulates a microRNA, and this is all this data is all in one flame retardant, IPP, which is an OPFR. We found where a gene regulates several different microRNAs, or we found cases where a gene or a transcription factor regulates itself. But what I wanted to show you today before wrapping up is the presence of these feed-forward loops in the flame retardant exposures. These feed-forward loops are cases where a transcription factor regulates another transcription factor, and together they both regulate a third gene. And these feed-forward loops are shown to be important in various disease mechanisms as well as maintenance of innate immunity, and may be important drivers of flame retardant toxicity and adverse health effects later in life. So just as a take-home message and conclusion, we showed that flame retardant regulatory mechanisms are not dependent on specific physicochemical classes. We also showed that microRNAs are important in regulation of flame retardant toxicity, and therefore they should be extensively studied. We showed that there are several common transcription factors that are also regulators of toxicity of multiple different flame retardants. Overall, the data helped us to identify common and unique gene clusters across individual flame retardants, but we show that it's still challenging to cluster flame retardants based on their genetic signatures and their chemical classes. But again, overall, these are all predictive relationships, and future steps should work towards functionally confirm the regulatory role of microRNAs in flame retardant induced gene expression, as well as some of the other targets that we identified, including these feed-forward loops. So acknowledgements. So this was primarily done with funding from a grant through the environmental US EPA, but also other fundings through NIH and IEHS, including our P30 Center. And my acknowledgements towards Dr. Tangwe, who is the lead PI on this project, as well as the different members of Tangwe Lab. And with that, thank you so much again for inviting me here today, and I will take any questions.
Great, thank you so much. So folks can raise their hand, they can put questions in the chat, or just go off mute. While we're waiting, I do have a couple questions. So I know it's difficult with the zebrafish embryos, but have you looked at maybe organ-specific changes in microRNA, or from the pathways, can you predict that?
Definitely, that's a really good question. From this early embryogenesis, the stages that we look at is around 48 hours post-fertilization, where organogenesis is at its full peak, and most organs are being developed during that life stage. So it's definitely particularly through gene ontology and other assessments and molecular biomarkers that are established for specific organs, it is definitely possible to look at organ-specific effects, for example, the liver or the heart or the brain, and then follow it up with more of functional studies, including staining and other approaches to look at specific organs. And one of the findings from this project was that, again, nervous system development is is affected, and we are trying to look at the brain functions and if there's effects on any brain functions that are related to behavior.
Great. Thank you. Peter, I saw you go off mute. Did you have a question?
Yeah, so I guess having different classes, you would predict that that if you use lower doses, I don't know if using lower doses would, and combinations of different classes of fire retardants would give you, will still maintain a fire retardant activity, but give you less toxicity. Is that something that you are looking into in terms of with combinations of fire retardants at lower doses give you less responses, let's say in your screen, or perhaps they may synergize in terms of their toxicity, especially given your concept here of the feed forward mechanism.
Yeah, that's a great question. One thing is that again, like this was a large scale screening that we did at EC80 concentrations to identify, to anchor the phenotypes to the gene expression changes. And once we do that and we have identified several markers, we can definitely do concentration responses. But also the other thing to just to focus on my initial method slide is that we see phenotypes usually at 5 days, whereas all of the molecular signatures or the molecular profiling that we do is at 2 days. And this is to eliminate any systemic toxicity that is derived from this, like overall toxicity when the embryos deform, or when obviously we are not profiling any dead embryos, but when the embryos deform, this leads to a lot of other systemic toxicity at 5 days. So our strategy is to avoid that whole scale systemic toxicity and detect the innate molecular responses that happen on the path to this systemic toxicity, which our hypothesis is that will give us the true molecular targets of that chemical that's driving some of the innate or functional effects. So at 48 hours, whenever we are profiling any embryos, all our embryos look totally normal. Many of these chemicals will go on to elicit morphological changes at 5 days. But when we are doing the molecular profiling at 2 days, the embryos look totally normal. So at that point of time, there are no gross deformities for the embryos.
Does that answer your question, Peter?
Yeah. Thanks, Subham.
Yeah, great. All right, we have a question from Rachel in the chat. Did you do any knockdowns or knockout studies for further investigation of the regulation of microRNAs or mRNAs?
That's in the works right now. So we are doing some of those studies, including some of the microRNA inhibitors and microRNA mimics, to look at specific flame retardants in the next in probably later part of this year.
Thank you. Carol, you have your hand up.
I was just trying to unmute there. Thanks for the great presentation. That was excellent. I have a couple of questions just about sort of technicalities of working with, put my hand down, the zebrafish embryos. Do you have to sequence a lot more deeply in order to see effects because you're working on the whole embryo? It sort of aligns with Connie's question as to tissue-specific responses. So now you've got this mix of tissues. Do you have to go really super deep then to see the effects you're expecting to see?
We definitely, so for most of our sequencing, we can detect enough of response or enough of the gene expression with around like 20 or even like 40 or even 20 million reads. In the past, as a part of my work at University of California, Riverside, and Connie's work, we have also used the Lexogen platform, which is a 3' sequencing platform with where per sample you can have lower number of reads, like 2.5 to 3 million reads, and we were able to detect organ specific or predict organ specific effects using this lower number of reads. So we don't have to sequence. Obviously, the more we sequence, the better. But definitely at that life stage, there's a lot of organ specific dynamics. But there's a lot, since most of the genes are active during those life stages, it is easier to detect a gene expression or differences in gene expression even with relatively lower number of reads. So we have been able to use, again, like around, like we have used 2.5 million reads per sample. We have used 20 or 40 million reads per sample. And in fact, like within our current lab, there's a project that is aimed at comparing where the divergence of prediction of have effects occur between between sequencing deeper versus sequencing less deep. But we are able to predict, even though it's a full embryo that we are homogenizing, we are able to predict effect using relatively less deeper sequencing.
Excellent, thank you. And the other question that I had was, you chose the 48-hour time point, but there's dynamics in the gene expression response, and there's different accumulation of chemicals, and reaching steady state at different times, or even accumulation. Do you think that you would get similar results, or have you done some time series to confirm that a longer exposure wouldn't increase the potency of some of these chemicals?
Yes, definitely. The longer exposure could increase the potency, which is why most of these embryos at 48 hours do not show any effects, do not show any morphological effects. But at 5 days, many of these embryos, particularly the ones which have morphological phenotypes, are deformed. But also with that, what comes is the whole systemic effects. So one organ being affected, the primary target organ being affected and shutting down the whole system. So our intention is to try to avoid that, which is why we select 48 hours. And definitely there's a lot of chemistry at play here, the depuration of the chemical, the metabolism of the chemical. So what we typically do is we do this whole on screening with a large library of chemicals. But once we have our chemicals of interest, and once we have our biomarkers of interest, then we do a time series, a temporal series. Then we do concentration responses that are focused on a few different chemicals.
Excellent. Thank you very much.
Thank you.
Thanks, Carol. And then Rachel put in the chat to connect with Maria Bondison. We've met her before at a few conferences, but it might be for the reporter specific for the organ niche question. That might be a good idea. Yeah, thanks, Rachel. You brought up in the beginning too, Subham, that microRNAs are pretty translatable across species, maybe more so than mRNAs. Have you looked in the literature to see if any of the ones you sequenced and then came up are appearing in other studies, or has that work not really been done in other species?
No, there's been work done in multiple different species, and most of these microRNAs, particularly some which we are focusing on right now with perspective of flame retardants, but also a few other chemicals such as pHs and other chemicals. They are also translatable to other species. They have the same seed sequence that bind that bind to like specific gene targets, specific gene sequences, or 3' UTRs of specific gene sequences. So most of these microRNAs, if not all, are translatable to even human or higher vertebrates.
Thank you. And are a lot of them embryonic lethal if you block them, if they're so conserved? Do you know about that?
Some of them are. Particularly, there's like for zebrafish, I know the really early stages of development, we have there's only one microRNA at play. I think it's microRNA 140, but I might be wrong. But that specific microRNA regulates the whole process of development during the early stages of development. So I will assume that if you knock out that microRNA, there will be lethality of the embryos.
Great, thank you. All right, I don't see more questions in the chat. Anyone else? You can either raise your hand or just come on. Giving people one last chance to hit unmute. All right. Well, if there's no other questions, we can all just thank our speaker again. This is a really fantastic talk. I think we can think about this within our microRNA working group, how we can apply some of these learnings too. And I just really appreciate it, Shubham. Thank you. And
Thank
thanks
you
to the
so much.
audience.
Thank you so much.
All right. Bye, everybody. Have a great rest of your day.